So this is where we were last time. You will recall that very briefly, I had just given you an overview of what phytoremediation is all about, because plants are also being looked at in a very promising way in order to carry out remediation of contaminated soils or contaminated water bodies. And here, very briefly, I had explained the four different mechanisms which have been understood to take part or to participate in the phytoremediation. And these were explained as phytostabilization, phytoextraction, phytostimulation or rhizodegradation, and phytodegradation. All right. So I had also explained to you now overall, whenever we look at the subject of phytoremediation, the way this subject is going to be covered is that the first thing is that before you begin about phytoremediation, particularly with respect to the heavy metal ions, you will see that the plants need to display a proper transport mechanism. So this is what we briefly started to learn in the last lecture. That is the first thing. How are these different heavy metals? They are taken up by the plants, or even if you talk about certain metalloids, etc. So what kind of transporters they lie within the root system, which allow these heavy metals to be taken up. And the second is, what is the basis? There has to be some kind of a biochemical basis for remediation. So today we will look at, for example, what are the mechanisms which are exhibited, which occur within the cell in order for phytostabilization to occur or phytoextraction to occur. Then I will also discuss with you certain case studies. And these case studies are going to let you know that there are practical examples where plants have been, where several trees have been planted in order to remediate the soil at the thermal power plant or at the mining sites, or tree like ficus, for example, can be used for phytoextraction, and similarly plants for treatment of textile waste waters. All right. So the coming few slides are going to be organized in this particular order. And the first thing is therefore the uptake of metals. And this is something which I discussed with you in the last lecture. I'm not going to repeat it, but I will just come to the underlying mechanisms, which are the biochemical mechanisms, which are exhibited in the root system or in the other parts of the plant for phytostabilization or phytoextraction. So what is listed in this particular figure are essentially, look at these numbers, number 1, number 2, number 3, 4, and what you see here, 5. So these are the 5 biochemical mechanisms which have been put forward. Okay, the first of these is, and the explanation is provided in this box here, the first is the binding of either metal or metalloids to the cell wall. All right. So obviously, metals, in order for them to be transported or to be taken up inside a plant root, and why we will discuss only about the roots here is because roots are the ones which are going to make a contact with the soil. Okay. So therefore, there are mechanisms which are operating within the roots, and that's what we are going to look at. So the first mechanism, what you see here, that these are the metalloids, etc., which can simply bind to the external cell wall here. All right. And how does it happen? It happens because of the presence of, for example, the organic acids which are constantly coming out of the root cell, because all root cells are going to give out what is called as root exudates. That is organic acids, and Na stands for nicotinamine. So these are the compounds which are constantly being excreted out of the cell wall. That is the cell wall of a root cell. So because these are negatively charged, if you look at organic acids like malate or citrate, for example, or fumarate, all of these are negatively charged in the soil, as a result of which they are able to bind to the positively charged metalloids, etc. So both nicotinamine or organic acids can participate in this process, and MT stands for metalloids. So it will just simply bind to the cell wall, as a result of which all the metal which you have now in the soil can be absorbed on the surface of the root tissue. And that is something what we call as phytostabilization, because now these metals are going to be no longer available in the soil for them to move from one part to another one, and they are simply immobilized on the surface of the root tissue. So that is the first mechanism which can operate. All right. Now, the second mechanism is chelation of metal with organic acids on the outside of the cell membrane. So many of these metals, along with these organic acids, etc., they can also pass through the pores of the cell wall and they can accumulate in this particular space. Okay, so you have now these metalloids which have chelated with these organic acids and they can stay on the outside. So on the what you have here is the cell wall, and what you have inside now is the plasma membrane. So in this particular space, many of these can be simply immobilized. Okay, so that's the reason why we say it is a mechanism which is exhibited in order to explain phytostabilization. So this will again will be classified as phytostabilization. So is that clear to everybody as to how it actually occurs on the surface of the root cell?
Yes, ma'am.
So that is the second. So mind you, still the metals or the metalloids, they have not been transported inside this cell. Okay, so everything, whatever happens right now, is either on the surface of the cell wall, or it could be in this space, which you also, you can call it as periplasmic space also. So they are going to be immobilized here. Or the third thing that can happen is that we write here as active metal. This is efflux. Efflux is to pump something out. Influx is if you want to take up metals inside the cell. So there are active metal effluxes which are operating within the plasma membrane, and that mechanism is shown here. And how does it happen? That is because you do have active transporters. So these are shown in green color. These are the active transporters which are localized along the plasma membrane, and just like any other transport protein which is membrane bound, it is also capable of taking these metalloids and it is able to transport them inside the cell. An example of the same you had seen in the last lecture. Okay, so for example, these are transporters which are normally for the root cell. It is involved in the transport of inorganic phosphate ion. However, the same transporter can also be used to transport arsenic. Okay, so these are active transporters which are located, and these are shown as green here. Okay, so you have these active transporters which can take up metals, transport them from the cell wall to the cytoplasm inside. All right, so when we discuss about the mechanism 3, it is because even if excess of these metals or metalloids are accumulated in the cytoplasm, there are efflux pumps which are capable of taking them and putting them outside the cell. So this is the third biochemical mechanism which can operate. The fourth is the metal chelator complexes. So if the metals are now through active transporters, if they are transported inside the cell, there are chelators which reside within these plant cells, which can be either PC, which are called as phytochelatins, or these are organic acids, or GSH, which is glutathione. Okay, so all of these are also capable of now chelating with these metalloids. So there are three categories of compounds as are shown here. And this is now, so they can be simply chelated. That is, now they will just reside within the cytoplasm as these complexes. They don't get transported. And or else what could happen is that there is a second category of active transporters, which are shown here in red. And these active transporters, they can take up free ion of these metalloids or chelates and remove them to the vacuole. All plant cells, they contain vacuoles, in which a lot of things which are excreted out during the metabolic processes, etc., can get accumulated in the vacuole. So in the green, what is shown is a plant vacuole, and either these metals or the metal chelates, they can directly enter inside. So this is a second membrane, which is a membrane which separates the cell or the cytoplasm from the vacuole. So these active transporters can take them up, and all these metals are now sequestered within within the vacuole. Okay, so the reason you see organic acids or you see glutathione is that you have the metal and the glutathione complex which has entered inside the vacuole, and it can stay as it is, or it can also be recycled. The metalloids are retained back, and things like phytochelatins or the glutathione can again come out of the vacuole, and then it can be recirculated. So therefore, it gives you an overview of what are the different kind of mechanisms which could be operating. Either there are chelators which are in the cytoplasm, or there are these negatively charged organic acids or nicotinamine, which is capable of binding to these metals and just simply absorbing them onto the root cell. Okay, so I hope it is clear as to, what kind of biochemistry is operating here.
So, ma'am, can you please explain the third point again? Ma'am, I did not understand why we have to, like, these metal wires have to go out.
Okay, see, because let me show you the last slide. I had also discussed because even in a, this is a schematic picture of a root cell. All right, now not only do they have transporters which are shown here, in which the direction is shown in this particular direction. Okay, that is from the outside to the inside. All right, so all the arrows which point inward are pointing towards the inside of the root cell. Apart from that, you also see these yellow colored pumps here, for example, and all of these are called as efflux transporters. Now, what it means is that they are simply dependent on the concentration of the metal which is accumulated. Okay, they only operate. Sometimes they can be active transporters. Otherwise, these are just operating on the principle of diffusion. That is, if you have too much of a metal which is accumulated inside the cell, like for example, the trivalent arsenic, then these efflux transporters will be able to pump out this extra arsenic out of the cell. And this simply operates on the principle of diffusion. The concentration of the metal is too much in the inside of the cell. So therefore, they just pump it out. So these are called as efflux transporters. And there are several of them. For example, here, this is still not an identified arsenic 3 transporter, or HMA5. For example, what does it stand for? It stands for the heavy metal ATPase. So this is an efflux transporter, but it utilizes the energy of ATP. It has to hydrolyze ATP in order to get energy so that it can now efflux out the metal ions. Or you have something here which are also a different category of efflux transporters. So these are routinely a part of the plants. Okay, because remember that metal is not always bad, because the plant cells, they need to take up metal ions. They don't take it up in the form of food items, and all that comes through the system of roots. So all the metals which are a part of so many enzymes, for example, copper, all your electron transport chain enzymes, they have copper, or there are so many enzymes which contain manganese as a cofactor or zinc as a cofactor. In their root, they anyway have these metal transporters. So do you understand? It's not something which is only for the purpose of bioremediation, but it is also because all the metals, they need to be taken up inside the plants, because it is a part of, it serves as a coenzyme for many enzymatic processes. So therefore, I mean, just as there are influx transporters, there are also efflux transporters. So that is how. So when we talk about these efflux of metals, it is already located within the root cell. And if the concentration of the metalloids becomes too much, they are able to efflux them out. So these are the various kind of biochemical mechanisms which the plants are going to use in order to take care of or to maintain a balance of certain metal ions. So the other thing is that, we also talked about a mechanism. Let me go back to this slide, which is the second mechanism is phytoextraction. Okay, now the last example which we saw was for phytostabilization, but the second mechanism which we had talked about was phytoextraction. That is, it is able to take up metals from the roots and transport it into the aerial parts of the plant. By aerial, what do we mean? That these are the parts of the plants like the trunk, for example, or the branches or the leaves, for example. So something which is in the air. So now the plant is capable, like, as you see here for phytoextraction, many of these metals are going to be taken up through the roots. They are transported here and eventually they can be deposited in the leaves. So this way, whatever as long as it is not toxic to the plants, it is capable of taking these metals from the soil, accumulating them in different parts of the plant, including the leaves. So that mechanism is called as phytoextraction. So in this slide is shown, for example, for arsenic, how arsenic can be taken up, how it can be transported, how it can be translocated into the leaves eventually, and detoxification. Because if you look at what is happening to the soil is definitely now detoxified because all the arsenic has gone into the plant. So the soil is now detoxified. Okay, so that part, you need to understand as to why we use the term detoxification. So again, here is a picture of a root cell. Yeah, any questions on this?
No, ma'am.
Okay, so here is a picture of a root cell. And what we are looking at here, for example, all of these are the different transporters and their names are also listed here. So for example, if I see something in red here, it is actually an arsenate transporter. In one of the previous slides, I also showed you that the normal transporter which uptakes the phosphate for the plants is also capable of taking up arsenate. Okay, so what you see here is the phosphate oblique arsenate transporters. Okay, so arsenic in the soil occurs in two forms. One is the arsenic which is in the pentavalent form, which is shown here, and the other is the arsenic which is now in the trivalent form.
Ma'am,
Okay, yeah.
arsenate is the iron.
Arsenic is a metal.
No, ma'am, arsenate.
Yeah, arsenate will be an ion. Arsenate will be an ion. Okay, but in which the valence of the arsenic is going to be, so it is pentavalent here. So not as a metal, but rather it is taken as a, it could be ASO4 minus. So in which case it is like an arsenate ion, and then it can be taken up by the same system as that, which is the phosphate transporter.
And ma'am, arsenate reductase is an enzyme which reduces GS to
Yes. Yeah,
GLS.
yeah. Let me take up one thing at a time in this.
Yes, ma'am.
There are several biochemical reactions which are occurring, but we will just take up one at a time. All right. So the first thing is, of course, the arsenate ion, which is pentavalent, and incidentally, it is, the trivalent ion of arsenic is not as toxic as it is the pentavalent form. So number one, that is something which you need to remember. Okay. Number two, both the pentavalent as well as the trivalent form of the ion can be taken up inside by two different transporters. So for the pentavalent one, it is the same as the phosphate transporter. But apart from that, there is also a NIP subfamily of aquaporins. Now, I had told you in the last lecture that water doesn't always enter into the plant cell as a result of diffusion. Not that there is too much of water outside and it can go inside the cell simply by diffusion, but no, there are pores in the cell wall which are called as aquaporins, which are capable of taking up these water molecules systematically inside the cell. Okay, and these are called as aquaporins. So what you see here in the yellow, it is that subfamily. So aquaporins, is a whole lot. There could be 10 families of aquaporins. One of them is a NIP subfamily. Okay, and that is the one which is actually used up for transporting the arsenate, which is the trivalent form. So there are two different systems of transporters. Okay, if arsenic is taken up in the pentavalent form, it undergoes a reduction immediately by the glutathione system. So here you have, so this is arsenate reductase is going to bring about reduction of pentavalent form into the trivalent form. So that is the first thing that is going to happen. So immediately there is a detoxification of arsenic by arsenate reductase, which are present in the cell. Now, you may say that how has this enzyme come? Because plants and soil, they have evolved in nature over a long period of time. So arsenate reductase is not something which has been introduced yesterday, but the plants, they already encounter, for example, in natural occurring activities, arsenate does get because of mining, because of other activities. this metal gets released into the environment. And over the course of evolution, the plants have, one of the enzymes, etc., could have mutated to make arsenate reductase. And since it gave a survival advantage to the plant, so it has been retained in the plant cell. Okay, so it's not something which is introduced into the plants through recombinant DNA technology, but it's an already existing enzyme in several plants, which is arsenate reductase. Okay, so then it will bring about reduction, and this gets oxidized. Glutathione gets oxidized. This accepts electrons, and now it becomes the trivalent form of arsenic. Okay, so that is the first thing which occurs. So detoxification, as you see here. And after that, this arsenate will combine again with the reduced form of GST. And now you see that there is a complex which is formed, and or it can also combine with these PCs, which I told you, which are the plastocyanins, which are plant chelators, which are already present in the cytoplasm, as shown to you in the last slide. Okay, so with this, they can be then in the form of, just like you saw in the previous slide, for example, that either in complexation with glutathione or in complexation with these phytochelatins, for example, it can be simply pumped into the vacuole. And the same thing which you see here also, for example, so this is a vacuole here. So it can simply, in any of these forms, it can now enter inside the vacuole. So this is a mechanism of phytodetoxification, because now they have just accumulated it in the vacuole, and as eventually the root cell, is not going to live forever. It has a certain life. And the root cell is going to divide, the new cells are going to take over, and the old cells, when they die, for example, all this metal is just going to be left behind in the vacuole. Okay, so that is how the detoxification can occur. Now, when you try to explain, for example, how phytoextraction is going to occur, that means translocation of this metal into the aerial parts of the plant. Okay, in order to understand is the second part. So the first step is detoxification, the second is sequestration into the vacuole, and the third is that of the transport. Okay, so both the trivalent form as well as the pentavalent, there are specific efflux pumps which you see here, for example, which could be the same as the phosphate arsenate transporter, or like the green one which you see here is a silicon arsenite efflux carrier here, or the blue one you see is an inositol transporter. Okay, through any of these, or then you see there is unknown, which is actually arsenic methylated, it's a transporter which can also take up the methylated form of arsenic. Okay, all of these will be able to pump it out of the root cell into the xylem. Now you will recall that in plants, especially, when you have all these trees and plants, there are xylem and phloem cells. Okay, so because that's like a carrier, it's like these are canals, which are vertical canals, which are going to transport through xylem, water is transported, and through phloem, you have all the nutrients, etc., which are transported into the upper parts of the plant, because the plants have all their nutrients in the soil. And they need to transport them through these vessels, which are phloem and xylem, into other parts of the plant. Okay, so what you see here as a xylem vessel is a vertical vessel which functions in this particular direction. Okay, so through that vessel, for example, once either the pentavalent or the trivalent form of arsenic moves into the xylem, it can be transported into that vessel, and eventually it gets distributed into leaves and other parts of the plant. Okay, so is this clear to everybody as to what is happening here?
Yes, ma'am.
Okay. Because till now it is just a case of phytostabilization. We have not looked at phytoextraction part. So this part of the picture will explain as to how phytoextraction can occur. So through the xylem vessels, all these, you see the red one and you see the blue one, the arsenate metal, the ions are going to move now, and then they are going to follow the same mechanisms as they did in the root cell. That is, these are going to be sequestered in the vacuole by interacting either with these phytochelators or with the glutathione, etc. And then eventually they will just end up in the vacuole of the plant. Okay, so this is like an isolated compartment, and it doesn't interfere with the photosynthetic activity, because all the photosynthesis occurs, remember, in the chloroplast. So that is again like a special organelle which is not exposed to these metal ions. So therefore, the photosynthetic activities are not compromised in the cytoplasm. All the biochemical activities are going on. They are not compromised because all the metals eventually will be sequestered in the vacuole. Okay, so yeah.
Can you talk about glutathione? I do not know.
Well, glutathione, you will recall, is a tripeptide. Okay, now I don't remember, but cysteine is one of the 3 amino acids. Okay, so imagine that glutathione, it only contains 3 amino acids. The central one is a cysteine. I think it has glutamate, then it has cysteine, and it has another amino acid. Now, when you say it is in the reduced form, then the cysteine amino acid exists in the form of SH. When you say oxidized glutathione, okay, so this SS refers to the disulfide bridge, which can be formed between 2 tripeptides. So one tripeptide, which tripeptide means, you just have 3 amino acids which are held together by a peptide bond. The central amino acid is a cysteine, in which the side group is SH. That is the reduced form. When you say oxidized form, then 2 of these tripeptides are going to come together. 2 cysteines will link together by a disulfide bridge. That is called as the oxidized glutathione, in which you have now 2 tripeptides which are linked together by a disulfide linkage. So is that clear?
Yes, ma'am. So when there will be sulfide bonds, it is in
exactly. So if we look at where my cursor is right now, when you say only GSH, that means the cysteine in this tripeptide is in the SH form, but in the oxidized form. Okay, because now it donates the electrons to here. And that is how now it becomes oxidized. So this becomes now, this becomes, oxidized, and this becomes reduced because it accepts the electrons, and that is how it goes from a pentavalent to a trivalent form. Okay, so I hope this part is very clear to everybody.
Yes, ma'am, yes. No,
okay, so you are accepting electrons. So obviously something is giving out electrons. That is the reduced glutathione. This is the oxidized form now, and this is the reduced form of arsenic. So, so that is, in other words, try to understand. It's also, very, very clear, self-explanatory slide, which will tell you as to how, for example, in the case of arsenic, not only do you have phytostabilization, but you also have phytoextraction. Because you have moved arsenic now from a root cell, you have mobilized it upwards through xylem, and eventually they end up in the aerial parts of the plant. Okay, so imagine if it's a very, very tall plant, it has enormous capacity to absorb these metals. So now, just as I had told you in the outline that, so we have looked at the uptake of metals. What are the mechanisms, like you saw just now, for phytostabilization, phytoextraction, and now we are going to do case studies. And here we will see as to how plants can be used to remove the wastes which are generated at the thermal power plant site or at the mining site. Then we will see how plants have been used for phytoextraction. This is particularly the silicon, the heavy metal wastes, etc. And also plants have been used. I mean, these are not theoretical, but there are, there are acres of land where plantation has been carried out in order to remove the wastewaters, which are colored wastewaters, which are generated from the textile industry. Now, first is that of the thermal power plant. Now, this is a problem which constantly occurs in those areas which are near where the thermal power plants are set up. Okay, so what kind of pollutants are expected to be there? Number one, so all the thermal power plants, they will use coal as a major fuel. So the plants will generate large amount of what is called as a combustion coal residues. Okay, so when coal is burnt, what kind of residues are left behind? These are fly ash, bottom ash, boiler slag, and flue gases. Now, what exactly do we mean by fly ash? So here is a picture to show you what the fly ash looks like. So these are the residues which are left behind when coal is burnt. So that's how it looks like. And then I also talked about the bottom ash. So this is what it looks like. And it's very different from fly ash, which is more dry. This is more mucky kind of a complex, kind of quite viscous. So bottom ash is left behind. Then there is a boiler slag. Okay, third category of compound, which is now called as a boiler slag. Okay, so all of these are left behind at the thermal power plants. In addition to that, also flue gases are liberated into the environment. Okay, now what is the composition of a flue gas? So first of all, when you look at fly ash, what is the composition of this fly ash or the bottom ash? It contains silicon dioxide, 60%. It also has aluminum oxide, 26%, iron oxide, magnesium oxide, calcium oxide, and potassium oxide. Okay, so this is all that it contains. So a lot of silica, aluminum oxide, etc. Okay, now in addition to that, flue gas composition. So we say flue gases are also liberated. Okay, now what is the composition of a flue gas? First of all, it contains all these nitrogen, carbon dioxide, water, oxygen. Apart from that, it also contains oxides of sulfur. That is because coal is basically it's like a fossil fuel only derived from the earth. So there are oxides of sulfur like sulfur dioxide or sulfur trioxide which are released, and you see it is percent ppm, 150 to 400 ppm. Similarly, carbon monoxide, it contains large amount of carbon monoxide, and we have already discussed that all of these are primary pollutants. Or oxides of nitrogen like nitric oxide, nitrogen dioxide, N2O, etc. Just as when you burn petroleum, all these oxides are released. Similarly, when you burn the coal, all these nitric oxides are also released. Okay, so all of these are actually bad for the environment. So apart from the air pollution, which we are already aware of, in addition to that, the soil also becomes contaminated with all these leftovers. And it is a major activity globally because that power, the major supplier of power, comes basically from the thermal power plants. The nuclear plants or other category, they only occupy a very small proportion of the power which is generated. Okay, so there is a lot of contamination there because the land or that area where plants may have been growing gradually will become totally plantless because all of these are very hazardous. Then apart from thermal power plants, the mining waste. So here is a picture. Yeah.
These fly ash, bottom ash, and boilers, like do we get them separated, or like these have been separated after burning this coal?
This is separated because eventually, whatever is left at the bottom of a boiler is also removed periodically, and it is left into the area which is surrounding the power plant because it doesn't get consumed. It has to be periodically cleaned up, and fly ash has to be removed from the plant, bottom ash, the boiler slag, etc. Then, again, these power plants are cleaned up and fresh coal is burnt, so on and so forth. Okay, so these are the kind of materials which actually come from burning of coal in a power plant.
Okay, ma'am. So, ma'am, like how it can be deposited anywhere, wherever we want. So, like, why are these becoming to that specific area only?
I will show you, I have a picture to show you as to, just let me come to the next and that'll answer. Because see, many of these things are also, as you will see, that they are put to use. It's not that they are waste and they are going to accumulate over a period of time, but there are several uses which these compounds they have been made to put to work. Okay, I'll show it to you in the next slide. But here is a picture, first of all, from...
One more question. Ma'am,
Yeah.
what is here in this slide on this left side you have written flue gas desulfurization materials. So what are these materials?
Uh, so you have, the boiler slag, the flue gases, and desulfurized materials. That is, apart from all of these, what we call as the combustion coal residues, etc. Now, coal also contains a lot of sulfur. Okay, so another component is called as a material which is desulfurized because you have the sulfur which has gone now in the form of oxides of sulfur. So some other residue is now called as the desulfurized material.
Okay, ma'am. Okay.
Okay, so here is a picture of where mining activities are carried out. And mining is a very big activity, especially in areas West Bengal, Jharkhand. These are very rich for so many minerals. Coal is one of them. Coal is also mined, but apart from that, ores, precious metals, like that's a huge industry. Aluminum, iron, and all the mining activities are carried out there. And this is what the landscape looks like after the mining activities are carried out. Okay, and our idea is to be able to restore these ecosystems to a previous picture. They should not look like this, where all the vegetation is removed. And the question is that whether plants can be put to use for restoration of these ecosystems. Okay, so now, for example, you just asked me, now fly ash uses. So the fly ash which is generated now, okay, from these thermal power plants, is not necessary that all of it is going to be accumulated. But as you will see, about 53% of it has found use. First of all, it can be used as a building material, as a backfill for road construction, for making pavements along the roads, for cement and brick manufacturing. It is used as a filler and asphalt mixture. It can be sometimes used as a soil stabilizer, as a cover suitable for soil reclamation or material recovery, for manufacturing of mineral wool and zeolite synthesis. Okay, so many of these, wherever something can be made use of, people have already found uses for it. But still, it's not able to take care of 100% of the residues which come out of a thermal power plant. Okay, so only, like when I give you this reference of the paper, you will see that about 53% of the fly ash can at least be put to different types of uses. Okay, so now before we look at anything else, just a little bit science to tell you that when you look at the physico-chemical characteristics of these fly ash, of course you saw what it looks like, for example. Okay, but then if you look at the physico-chemical characteristics of this, it contains, for example, sand. Okay, 56 to 82%, it contains only sand. That is what the material is, which is actually silicon oxide. Okay, like you saw in the previous slide. Okay, so we said when you look at the fly ash compounds, silicon dioxide makes up 60%, aluminum oxide makes up 26%, and silicon dioxide, it's nothing but sand. So there are three columns which I just want you to look at, just to allow you to appreciate as to what you are dealing with. Okay, so if it is unweathered, unweathered is something that means the fly ash has just come out of a thermal power plant. That is called as unweathered. That is no weather treatment. By weather, what do we mean? Rain, wind, lightning, and so many other factors. When they fall on these, then the sand becomes weathered, in which case the composition of these things can also change as a consequence of weathering. And that is why we use a term like either the unweathered fly ash, or we say the weathered fly ash, for example, or the mining wastes. All right, so if you look at the sand percentage, it's very high in sand, 56 to 82%, which is reported by this group. Then in Serbia, in some other where they are using coal in the power plant, they have reported up to 97% of sand. Okay, and this is the composition in the weathered fly ash. This is the composition of sand in the mine waste. So that's the way you're supposed to interpret. Similarly, silt. Now, silt is nothing but slightly finer sand. Okay, sand is more in terms of particulate matter, in terms of the nanometer, the size of the matter, it is much bigger. Silt is a little bit lesser, and silt settles also at the bottom of the lakes very easily. It's like a fine sand. So here is a composition, for example, which is given of all of these materials. Now clay, clay is something which is a little bit softer material, because with clay, you can the earthen pots and other things, because it can bind. Clay is a little bit more refined, and once water is added to it, then it becomes more like a plastic, and that is why earthen pots and other things can be made out of clay. They cannot be made out of sand. Okay, so clay also exists. That's also present in the fly ash. So that's a much finer composition. So 1 to 4% or whatever. In addition to that, hygroscopic water, that is by virtue of the property of the material, how much water it is able to retain. It's only 0.54%. Because you can imagine that, sand doesn't hold water, silt doesn't hold water, clay holds a little bit of the water. Okay, so very, very less amount of this water it is able to hold, because if you put water in the sand, it immediately goes through, and that's not a good quality for a soil. Because in any soil, if vegetation has to occur, if plants have to grow, it should be able to hold water, not just go into the groundwater. Okay, so that means by hygroscopic water, it is a measure of water permeability. So all these materials have great water permeability. They just don't hold water. If you look at the pH, for example, the pH is on the alkaline side. And what does it do? It enhances the release of the metals. That means the metals are not going to be surrounding the soil or the roots, as a result of which the plant which depend on all these metal ions for their survival, they are not able to take up metals, because metals are, simply they are just blown away with alkalinity. They don't stay in the soil. Okay, then they also contain very large amount of soluble salts, as a result of which the electrical conductivity goes very high in the soil, which is not a good thing for the soil. Okay, so I hope all of these points you are able to look at and pay attention to, what is the purpose? You have looked at only those properties, but in terms of, what are the chemical properties which are altered as a result of these is given here. The carbon content is okay. It's not very bad, 3%, but the nitrogen content is very low. The potassium, because all of these are, major nutrients for the soil. Okay, so all of these are summarized in this here. So whatever you have seen here, these are limiting factors for plant growth. What is it? The mechanical composition you have seen, that is sand, silt, etc. The pH is unfavorable. There is a high concentration of soluble salts, as a result of which electrical conductivity, lack of essential nutrients like nitrogen and phosphorus. They can also contain toxic concentrations of various metals. They also reduce the number of microorganisms which can fix the nitrogen, and they also reduce presence of mycorrhiza. Now mycorrhiza, myco means fungus and rhizo means roots, because in a normal scenario, the roots are also associated with very special category of fungi. It's a symbiotic relationship in nature. Okay, so because of all of these properties, there is lesser fungi are going to be associated with the roots, and that is going to be a disadvantage for the plants. Or it is a factor which can limit the plant growth. So is this point clear? So please have a look at this table in order to understand as to what is the importance of it. So is this clear to everybody?
Yes, ma'am.
And what about other students? Please, are you listening? Are you attentive in the lecture? Okay, so the first four pictures here, which are like A, B, C, and D. All right, these are where the thermal power plants have been set up, and it is from Serbia. So the fly ash deposits, you see all of these here are the fly ash deposits. at the thermoelectric plant station, which is Nikola Tesla A in a city in Serbia. Active cassette of fly ash. Okay, so whatever you see here, for example, in pictures A, B, so these are all, you know, fly ash deposits which are running in between these plants, and large areas here, they have fly ash deposits, or what you see here. And this is the power plant. Okay. And now what has been done is that systematically, obviously, you know, the local vegetation has to be seen as to what kind of plants can grow at that latitude, at that altitude, etc. So what has been done is that gradually, you know, plants have been deliberately sown over this area. So what you see here in E, F, G, H, and I, these are the passive cassettes of fly ash 3 years after bio recultivation with sown legumes, grasses, and shrubs, etc. Okay, so all of these have been actually plants have been planted there deliberately. And 3 years after the plantation was set up, the area looks what you see here in these particular pictures. Okay, and then JKLMN, what you see, these are all passive. That means that there is no more fly ash which is being deposited in these areas anymore. So these are called as the passive cassettes of the fly ash 11 years after biorecultivation with spontaneous colonizers. Okay, now the difference is that initially, you know, the conditions are not ideal for these particular plants to grow in this area. Okay, so there are only special classes of legumes, grasses, and shrubs, etc., which can be planted in these areas. And naturally, there is a natural succession which occurs. Okay, and then gradually these are going to die out and something else will take over, some other kind of vegetation, etc. will take over. So 11 years after biorecultivation, so you see now that, you know, something, and even they may have put some plants which are brought from outside and they have planted it on the soil because now the conditions are suitable for these plants to grow here. Okay, so here is, you see something which is now you can call it as eco restoration. So what is eco restoration? It is a process of renewing the ecosystem stability and resilience after stress or disturbance with respect to its health, integrity, and sustainability, etc. Okay, so please, so are you able to see what is the practical application of phytoremediation?
Yes, but we said in the last slide that these materials have only 0.54% of water content. Like they can keep only this much percentage of water. So like how can this has been grown at the first place, like at C part and D part?
Yeah. Yes, you know, these areas have not been greatly, or this set of crop is not greatly affected the way it is affected here, for example. Okay, here, of course, you see that on the periphery, maybe the concentration of the silt and other things is much lower, and that is why some plants have grown up. Because, you know, I mean, this is a reality that in the sense that at the periphery, you do see that some plants are still capable of growing, whereas in this particular area, it doesn't grow. And this could be slightly, you know, located a little bit upstream from where the actual power plant is located. Okay, so what do plants do? They stabilize. So again, it is phytostabilization. They can phytostabilize the fly ash and something which comes from the mines, etc. They also prevent wind erosion. Okay, they decrease the mobility, the toxicity, and the dispersion of chemical elements in the surrounding environment. Then plants also provide the organic substances, like, you know, we looked at the organic acids and so many other exudates, or the plants will bring in mycorrhiza in the plants, like, because there are certain legumes also. You see here, the biocultivation has been carried out with some legumes. So legumes are already nitrogen fixers. So as a result of which the soil becomes now enriched in the nitrogen part. And grasses, you know, are photosynthetically more active. All grasses are C4 plants. So their photosynthetic efficiencies are much more. The grasses grow, you know, in terms of the total biomass which you get per quanta of sunlight which comes there is much more compared to that what you get in the dicotyledon plant. So more green matter means more organic matter, etc. So all of these, all of these basically can be used and can provide a practical solution to remediation of such kind of areas. And then, you have, for example, this picture I showed you earlier also, that due to mining activities like here, it's a picture of coal mining. So what you see that there is a large change in the landscape. Earlier it was like, like semi-green, or there was some kind of a green cover here. But now when you do all this mining, obviously, everything is exposed here. So there is degradation of land, deterioration of air and water quality, near total loss of vegetation, affecting negatively fauna and human beings. And all of these waste rock and tailings, they contain a lot of metalloids which come out of the earth, persistent organic chemicals, etc. The fine particles, they also cause irritation in the eyes, etc. And lead to many other things for people who are either directly working there or for the areas which are adjacent to them. So please, is this clear to everybody?
Yes, ma'am.
Okay, so I'm just going to take a 5-minute break. We are at 10:26, so 10:31, I'll just come back, and just a 5-minute break for all of us.
Yes, ma'am.
Okay, so are the students back?
Yes, ma'am.
Okay, okay. So now let me go over to the next slide. And so basically, I don't want to flood you with too much of information. But when you talk about mining, there are some important like words which are used and which you need to understand. And one of them, for example, so all the waste which you get from the mining is categorized into three different sections. The first is called as an overburden. Okay, the meaning of overburden is that whenever you carry out a mining activity, the earth is going to be dug up. Okay, and it is not necessarily the ore part, but it is the earth, the subsurface soil, etc., which is now left on one side before the mining or before other activities can be carried out. That part of the soil is called as the overburden. The second is called as the waste rock. That is, this is like a second layer of soil from which you could have taken out, for example, because if you talk about ores and other things, there is a low quality ore which you don't want to carry further for processing, etc. Then there is a high quality ore. So it is a high quality ore which is going to be carried forward for processing, but the low quality, etc., is now called as a waste rock, because the concentration of the metal is so low in these rocks that you don't want to process them any further. That part of the waste during mining is called as a waste rock. And finally, you have the tailings. That is, once the ore is processed, once the metal is extracted, like, you could have an aluminum ore, you could have an iron ore. And once iron is extracted, there is something which is now left behind. Okay, and these are called as the tailings, which could be either, it could be the coarse fraction or it could be the fine fraction. Okay, so that is how the mining wastes are categorized. Now, what do you do with them? The first thing, the overburden, that is the soil, etc. So this overburden can be used for revegetation during the mine closure. So once you close the mine or the mine becomes inactive, the soil, this is taken, and then again it can go back into the same pit. That is called as revegetation during the mine closure. The waste rock which you see here, for example, this can be reprocessed to extract the minerals and metals, and it can be used as a backfill or an aggregate for road construction. And then you have what you call as the tailings. So rich tailings like manganese and clay, they can be used in agro, like, if you are mining manganese, for example. Okay, so this manganese is a very important nutrient for the plants also. Then it can be used in agroforestry, because it can go for enrichment of manganese in the soil, or it can go for construction material like bricks and cement. So whatever is left now needs to be discarded. So this is discarded now into ponds, into surface waters, and into groundwater. Okay, or many of these will be just heaped up, or it can go into underground mines or open pits, etc. So this is the fate of the different things which are being released during the mining activities. So here is an example to show you how, like, reforestation, or you can say that plantation can now be used in order to carry out remediation. So in the first A to C, A, B, and C. Okay, so let me read out the legend. These are the mine waste sites in Serbia, copper mine waste in metalliferous bore region, etc. So in A to C, these are the mine wastes which you see. Okay, so they are all collected in heaps, and they are just collected on one side. They are all like piles are built up along the water bodies. They are simply piled up here, and this is what it looks like. Okay, and now, if you have, for example, plantation of, this is some kind of a plant which is shown in D. So some kind of a plantation has been carried out on this particular site. So you see some kind of a green cover is provided. And in E, there is another type of a plant. And similarly, in F, there is another type of a plant which has been planted here. So this is just an effort in order to do, restoration of the ecosystem here. Similarly, what you see in G and H here, for example, these are the mine flotation tailings at the lead, copper, zinc mine in some area in Serbia. Okay, so in G and H, because copper is mined here, so you see that some of it also appears pink in color. And similarly here also, these are some of the wastes which are deposited from the mining industry. And then after some time, there are these colonizer, which you see the small plants which have been planted here in this area. Or you see that something else. This is antimony flotation tailing here after some flooding event has occurred, and 1 year later. So you see that, for example, lot of plants have started to grow here, or they have been actively planted in these areas. So the idea here is only to allow you to understand that microbial consortium is not the only way that ecosystems can be restored. So here is an example where trees, etc., can also be used in order to restore the ecosystem, and that is why we call it as a phytoremediation. Okay, so if this is clear to everybody, then I'm going to do another case study with you.
Yes, ma'am, it is clear.
All right.
And we can see like phytoremediation is faster than microbial remediation, right?
It'll be slower, but it is something which you don't have to bother. You don't have to replenish it every year, because once during the initial stages, maybe the first 4 to 6 months, you may have to take care of the plants to be able to provide them with fertilizers or organic carbon and other materials. But then once the plants will establish themselves, then the natural process will take over, and eventually the area can be reclaimed. However, it is going to take years for that to occur. It could be 5 years, or like you saw in that other case, that 11 years it took for that area to be rehabilitated where the thermal power plants were there. Okay, so it's a
Yes,
slow
ma'am.
process. It's a slow process, but then it is possible. It's not that the land has absolutely gone bad. Okay, so now let me give you another very interesting example, which is a case of phytoremediation, where heavy metals, etc., can be removed. And the case study here is of an area in China. And so basically, apart from the mining and other things which we have already talked about, there are a lot of heavy metals which keep
Ma'am,
accumulating. Yeah.
we are not able to see the slides. It is the a slide. It's written importance of vegetation cover.
Maybe
Perhaps you have changed the slide.
I have changed the PPT, so I need to go back, and I think I need to, yeah, take that other one off. Yeah, just give me a minute. Yeah. Okay, so can you see it now?
Yes, ma'am. Now it
Okay.
is visible. Lecture 16.
Okay. So this is another example. Okay, and of course, this is the details are from the paper, and I'm sure you will quite appreciate, and you will also see, how much more can be learned from these systems. So let me project this slide and explain it. Now, there are a lot of heavy metals which keep accumulating. Okay, and even in higher organisms, and this is through food chain. So I don't have to tell you that, what is the source of metals in the environment. So there are several natural sources of metals, like due to volcanic activity, due to weathering of the rocks, due to several processes, which are associated with the rainfall, with carryover of these saline water intrusion, etc. So these are all natural sources of metals. Then there are the human activities, like metal smelting, gas combustion, construction activities going on, refuse incineration, electronic wastes, etc. So these are all man-made activities which we constantly do, as a result of which the metal load in the nature increases. Or it could be man-associated activities, not necessarily related to industry, but for example, natural activities. So sometimes there is littering, you carry out enrichment, you take soil from one place to another one, so on and so forth. Or there is electronic wastes which are constantly dumped, for example, which bring in gold, platinum, cadmium, lead, strontium, chromium, etc. All of these are also released. Okay. Now, one such site, especially, the emphasis here is on the electronic wastes, because today, all the mobiles and the laptops and what have you, all of us are using them heavily. But then what happens eventually? It doesn't get recycled. Okay. Now, globally, unfortunately, some of the poorer countries, including India, are the ones where a lot of illegal trading is occurring of this silicon waste, okay, of electronic waste. And as a result of which, and China also, and many of these Southeast Asian countries which are very poor, they have been used by rich countries for dumping of electronic wastes. Okay, so one such place is in this town in China, where there is dumping by US and also in the European Union. Electronic wastes are illegally dumped into this township in China. Okay, so they have been facing a major problem of invasion. So here is a map of China. Okay,
Ma'am,
and yeah.
can you go back to the last slide? Ma'am, what is pedogenic processes? Can you explain again?
Yeah, pedogenic is also, these are man-made activities. Okay, but uh, but these activities, they also involve uh, addition. That is, uh, like when people go across, there is a lot of littering that they carry out. Okay, now it is not something which is related to industrialization. There we know human activities, like you have the painting industry, or you have the mining activities which are going on. So these are man-made activities, but nevertheless these are like littering, for example, or you carry out soil from one place to another, transfer it, or enrichment, certain areas, soils, etc. are going to be enriched. And this is, I think, burning, etc. Sometimes the soil, etc. are burnt by humans. Okay, like deforestation and other activities, or there could be natural. So all of these activities are called as pedogenic activities, and they're not directly related to industrialization processes.
Yes, ma'am, understood. Also, like how US is sending this all this waste materials into China. Like they have to export, and Chinese will they are not checking it that wastes are coming. What are
Absolutely. So, this is true for all, I think, although China is a developed country, but nevertheless, because of large population, I think everything is not as systematic. There is a lot of illegal activity also which goes on there. So some of these, for example, so this particular town which you see here, Guiyu, is one such town. And similarly, this Longtang is another town, Dali. So many of these towns are identified here, which have essentially become the dumping grounds for electronic wastes, both from the US as well as from the European Union. Okay, it has been estimated that approximately 9 million tons of e-wastes, of which 10 to 15% are illegally dumped into China. Okay, now, of course, apart from environmental hazard that it creates, because all of these will contain heavy metals, what else is there? The negative impact. So in the European Union, for example, what is the negative impact of this illegal dumping in China? Firstly, there is an economic impact. Why? Because it is a loss to the recycling industry. Because if huge tons of this electronic wastes are now being moved out of the country, I mean, there is an e-cycling which is operating within their own countries. So that is going to not benefit because of this illegal training. That is the reason why we say that it has a negative impact on the economy of that place. There is an economic loss to the recycling industry, and also there are job losses. Why? Because people will be associated with this e-recycling and they are not able to get job. Then there is also a social impact. That is, there is a negative impact of this is an organized crime activity which has a social impact in the European Union. Okay. But it has a positive impact in the European activity, especially for people who are involved in these illegal activities. Now, what happens in China? Again, there is an environmental impact. So there is, there is other things could be there. It may provide extra jobs to the lower strata of people, but there is a huge environmental impact, because soil, groundwater contamination, air pollution, etc. occurs because of dumping of the e-waste. There is human health impact, respiratory problems, reproductive problems, injuries, etc. There is a social impact. Child and women workers, mostly these are these are not really in the recognized domain of work. There are no social benefits to these people, low wages, etc. So it has a bad social impact. There is an economic impact, economic loss to the formal recycling industry. Okay, so just to give you, so it is not only about the environmental part, that when you talk about the e-wastes, it has a societal impact and it also has an impact on the finance part. Okay, so now in this study, it is a paper which is on heavy metal remediation using this tree, which is Ficus microcarpa, through transplantation. And then let's see these sites which have been used for over years as dumping sites, whether trees can be planted in there and whether that area can be restored. Okay, so this piece of work was carried out by this group of people, and it's also a part of a published paper which came out in 2018. So this is what the tree looks like, ficus. It is generally an ornamental tree and it can also be used as a bonsai. Many people keep this ficus. Also, they just make small plants and it can be used as a decorative plant. So we will see in this study how ficus has been used for carrying out bioremediation. So this is an actual picture of this is the dumping site you see here. Okay, and it is surrounded by, there is a play field here, a soccer field in this area. Okay, and this is where some of this e-refuse is burnt. So this is the main area right here. And then there are people dwelling area, there are people who are living in these zones, so on and so forth. So this is just a Google of that map of that particular area. So what was selected now is there is an area of 400 square meters which is situated at the transition zone among the agricultural land, the e-waste dismantling site, and the open burning field, which has been now selected for these experimental sites. Okay, so somewhere like here, this is the area which has been selected for planting these trees. So is this clear?
Yes, ma'am.
Now, what is the experimental scheme? Okay, how can this eco restoration, or how can plants be used? So what is done was that these plants are taken, which is Ficus microcarpa, which have very small plants, with a very, so before I give you the plan of experiment, let me show you a picture. Maybe I should have shown it to you first. So essentially the theme of this experiment was that this was a polluted soil which contained a lot of these heavy metals from the e-waste like cadmium, copper, mercury, lead, etc., which are found in these soils. Okay, so the idea here is that a tree is going to be planted as a result of these phyto extraction or phytoremediation. Many of these are going to be taken up by the plants, and you will remove the plant after a period of some time. The plant is taken up, so the soil is now going to be depolluted. Soil is going to be cleaned up, and this particular tree is now taken and it is replanted in some other area into a clean soil. And eventually, either it retains all of these heavy metals or it moves it into biosphere. So then you are going to measure the metal concentration in the bulk soil, in this clean soil, in the rhizosphere and in the plant parts. Okay, so what you have done is that in the polluted soil, initially you plant trees and you allow all the metals to be taken up by this plant. So this particular soil becomes clean. Then you take these plants and you plant them elsewhere at a second site, which is called as a clean soil. So some of the metal ions which have been accumulated by the plant are going to move into the rhizosphere, and then you measure what is the metal concentration in the bulk soil, which is, in this larger area. Rhizosphere is the soil which is very closely associated with the roots and in the rest of the plants. So that is what the objective of this experimental scheme was. So is that clear to everybody?
Yes, ma'am.
Okay, so now let me go back and very quickly give you the experimental scheme. So that is the way it should be understood, that this ficus plant is taken, similar morphology. Obviously, you don't want any variation in the beginning. So you make sure that you select the plants which are approximately the same in height, the diameter at the trunk, the height and the crown, etc. They are now cultivated on the chosen 400 square meter field with a low density. So you put about 2,500 plants per hectare of land, so that there is no interaction between the plants. Otherwise the plants are also going to compete for the nutrients. So you plant them at sufficient distances from each other. So 2,500 plants were now planted per hectare. That was the density. So 4 subsamples were quickly collected, and then approximately 15 to a depth of 15 centimeter. So you dig under the ground up to 15 centimeter. After the plantation is done, then this is going to serve as a control sample for you. Okay, so within a 50 centimeter radius around each plant, you simply take out this particular plant and it is mixed thoroughly to constitute a composite sample. And this sample is air dried and it is sieved to remove the foreign material and the coarse particles. And entire plant is now uprooted and it is classified according to the pollution level, etc. It is cleaned up, etc. Then a part of the root, stem and leaves, etc. So this is all at control time. Okay, so at time 0, for example, the root, stem and leaves are cut, they are sampled. The epidermis, that is the outer layer of the plant, is taken away from the roots, and the remainder is cortex and steel. And so all of these results we are going to discuss. And then what is important is that at this particular site, the plant is allowed to grow for a period of 3 months. And after that, the plants are removed from there, they are separated, and so on and so forth. And then they are replanted again at some other location. So is this clear? So in the initial part, then it is removed and then it is replanted. And then it is allowed to grow also for a 3-month period. Okay. So now in this particular land, the first thing that is done is that, you have first the soil characteristics. What is the distribution of the heavy metal in this 400 hectare land which they have chosen? Okay, so this particular picture shows you the distribution of cadmium, that is milligram per kilogram of soil, which is taken from this region. All right, so the crossed region, what you see here, contains, for example, if it contains more than 0.6 milligram of cadmium per kilogram of soil, okay, then it is called as, so this is heavily contaminated soil, heavily polluted soil. And the 18 number indicates that if the total cadmium content is so much, this is 18% of the total cadmium content is distributed only in this particular area. So I hope you're able to understand all of these numbers.
Yes, ma'am. Yes.
Okay, because there are 3 different zones here. This is the zone where the concentration of cadmium, as it explains here, is less than 0.3 milligram of cadmium per kilogram of soil. And that region is shown like this. And then you see that in this particular region, for example, the concentration of cadmium is between 0.3 to 0.6 milligram per kilogram. And this makes up for 43% of the cadmium, which is in the entire area. This makes up 18%. And of course, the remaining is 100 minus this plus this. Okay, so just try to understand. So this is what, and concentration here is less than 0.3. In a similar manner, there is also the data which is collected for copper. Okay, so in this area, the concentration of copper is less than 50 milligram per kilogram. And in this area, the concentration is between 50 to 100 milligram per kilogram of soil. So this is like mildly polluted, and this is the least polluted. this will be the most polluted because here the concentration is 100 milligram per kilogram, but it only makes up 3% of the total soil. So this is your experimental site where you are going to carry out plantation of these trees, ficus trees. So now the data which you see here, for example, you see 3 different categories. So the clean category, remember there were 3 different categories in the previous picture. So these refer to exactly the same way, either the clean area, the polluted area, or the heavily polluted area. N refers to the number of plants which have been sampled. So in the clean area, the data which you are looking at, that is either the soil metal content, or you are looking at the root biomass, you are looking at the shoot biomass. All this data is gathered for a sample number which is 39. So 39 plants have been taken from the clean area, 43 have been taken up from the polluted area, 18 plants have been taken up from the heavily polluted area. So what is the range of the soil metal content? So if you now try to measure, okay, what was the soil in this particular area, how much of metal it had, okay, you will see that the range was 0.04 to 0.29 milligram per kilogram of soil. Okay, and in the root biomass at that time it was 4.99 cadmium. The shoot biomass is 7.44. The whole biomass, that is root plus shoot. So this is the total amount of root, which you are taking, total amount of shoot which you are taking, and this is how much of soil. So, so this soil metal content which you see is not really what is in the root, what is in the shoot, because in the early on in the experiment, you don't expect these heavy metals to be either in the root or in the shoot. So the data which is presented here is only what is at time 0. What is the amount of metal which is present at time 0 in the soil?
Yes, ma'am. And after
and then
what is the duration after which this root biomass and shoot biomass has been measured? After like what is the duration of this transplantation?
Yeah, that is, so this is the initial root biomass. If you have in the control sample, because you made sure that all the plants which you have taken are going to be roughly of the same size. So at time 0, the total, if you have 39 plants, okay, and you just cut the leaves, you cut away the stem and you cut away the root, and then you weigh, then you see that 4.99 gram of root material is there and shoot is so much of material. So this is all at time 0. Okay, I hope you're able to understand. So let's stop here because we are already running out of time. It's 10:59, it's already 11, and many of you will be having. So we will come back in the next class and we will finish up this lecture.
Yes, ma'am. Ma'am, I had a request, like, can we have our quiz, like, after 14 offline? That will be better, like, and you should also give us, like, our term paper topics, or we have to choose the term paper topics, and what will be the deadline for the term paper topics?
Term paper topics also, maybe I will just circulate a list and people can give me their choices. I will suggest some topics, maybe 20, 25. And other people, if they want to come and suggest a title of their own, even that is fine with me, but provided you show me what papers you are going to consult, because you should read.
Before coming to term paper, ma'am, I think that quiz should be offline, ma'am. Can you take it offline?
Absolutely. Yeah, I would love to take it offline. Look, it depends on you people whenever, because I don't want to move. The semester is also very compressed. First is our last day, and I don't know how many buffer days we have in, or we could just do a half an hour quiz in the class. I don't mind.
Yes, ma'am. That is.
Okay. So you are expecting everybody is expected to return by 14th.
Yes, ma'am.
Isn't
so
it?
After 14, you can take the
after 14, and I'm sure
the
classes. So then what we could do is that in the week which is following, 14th is actually a Monday. So maybe the week of 21st we are going to keep our quiz. Maybe 21st we can keep our quiz, and I'll book a classroom for that.
Yes, ma'am. That is perfectly fine.
Okay.
Yes, ma'am.
I'll just send out a mail to everybody that in the class, which will be the 21st class, we will all assemble in a classroom and we will have our quiz. Of course, class, I don't know, where do I go back or I can continue to teach? If most of you turn up, I can continue to teach in the class that day.
Yes,
Also.
class would be really fine.
Yes,
Okay.
ma'am. Classes are also fine if it will be offline.
Yeah, yeah. Okay, let's see what's the response like. Okay then. Okay. I'll
And the
see.
major will be also offline.
Quiz will be offline because that's what seemed. I know because that's the only way you are going to study.
Yes, ma'am.
Okay.
Ma'am, what is the
Yeah.
syllabus for the quiz?
You see, I think microbial biosensors we have finished. After that, we did the biogeochemical cycles. We are doing phytoremediation, and we will be able to do one more topic, and maybe it is energy and recovery from the waste. That is how methane can be produced, and they're also interesting. There are just 2 lectures in there, so we should be able to finish it, I think. So there will be 3 topics which will be part of the quiz.
Okay, ma'am. So the part of the
Ma'am,
syllabus
the
after minor.
After minor, yeah.
Okay,
Don't
ma'am.
let's not bring up anything now. I'm just focused so that all of you can do well in the quiz.
Okay, ma'am.
Okay.
after minor till the class 21st.
After minor until whatever we do up to 21st.
Yes, ma'am.
Okay, okay, fine. I'll just send it out, on the team. So I'll just send out a message to everybody so that people are also mentally prepared.